Hello everyone, this is Elisabeth and I'm Annalena. And this behind us is Kaarina, Professor Dr. Kaarina Sivonen. She was born on the 23rd of August 1954 and her zodiac sign is Leo. They tend to be powerful, intelligent and inspiring. She was born in a small town in the middle of Finland called Eötvös. Her parents lived there in their lifetime. Now she uses her childhood home as summer vacation place, and it also has a nice sauna.
First, she studied environmental conservation, trying to save the world. Later, she studied microbiology. In her PhD thesis, she investigated the occurrence of toxic cyanobacteria in Finnish lakes and the Baltic Sea. She's a professor in microbiology and head of the Department of Microbiology. Her main tasks are teaching, supervising, administration, and research.
As a child, she loved reading a lot, so writing stories seems like a perfectly fitting dream job to her. A fun fact about her is that she's a bad swimmer, though she loves snorkeling. She once was invited to Heron Island in Australia to a conference where she also went to the sea. She said snorkeling in the reef was awesome.
She's most grateful that she's healthy, at least to her knowledge, and for the people around her, as her husband, her relatives, friends, and good colleagues. She won too many prizes and awards to list them all, but among them was a Science Award from the Helsinki city for excellent research on cyanobacteria in 2001.
She reads a lot, but tries to move too, skiing in winter, hiking and picking up berries from the forest in summer. But she also loves her own garden very much. Her most beautiful childhood memory is being together with her family, playing with her siblings and visiting her grandparents.
Her friends would describe her as reliable and having a good sense of humor. She likes to laugh a lot. She also has a pet. As you can see, it's a cat, and its name is Siri.
Every day is a new opportunity, finding new interesting results and meeting nice people. That's what motivates her to get up every morning. When it comes to music, she likes all kinds of music, from opera to blues or pop music. But as an example, she likes the song of Adele a lot.
Her favorite reservoir is the lake nearby her childhood home. Her biggest success was finding how common toxic cyanobacteria are in our lakes and in the Baltic Sea, solving the structures of the toxins, finding the producers, and based on biosynthesis, developing detection methods for toxic cyanobacteria. Please welcome now Professor Dr. Kaarina Sivonen.
Good morning, everyone. Nice to see you all. And I would like to thank the girls, very nice introduction, and then also the organizers inviting me. I was once invited to South Korea, and I was the only female and only European, so I was very honored. But now I'm honored to belong to these 5 persons here. And my title is Cyanobacteria in Water Ecosystems, but I'm going to speak mostly about the phenomenon you can see in the background, mass occurrences of cyanobacteria. So mass occurrences of cyanobacteria can be problematic. They might be aesthetic problem, the water doesn't look so nice, and here you can see, here you can see this bluish color. These cyanobacteria growing here, they are lysing and liberating this cyanophysin pigment, which gives this bluish color to these organisms. They also might produce odorous metabolites such as methyl isobornyl and geosmin, which gives a kind of muddy, earthy odor and tasty dish. And we know now that they can be also toxic, which is a health risk for human beings and animals as well. And the major toxin types are these hepatotoxins and neurotoxins. So many years back, when people wanted to know if there are toxic cyanobacteria, the sampling was important, and here you can see Baltic Sea, there is Scotland Island, there is the Baltic countries, Sweden over here, and you can see that cyanobacteria can be seen from the satellite as well, so they are not small. And I have to tell an anecdote. There is this research vessel we had to use while doing these cruises in the Baltic Sea. But when I went first time to this cruise, I got so seasick. After that, I have sent my students to follow the sampling. So here are the table of surveys, systematic surveys made in Europe, in European countries. I made one of the first ones in Finland, and we found we analyzed over 200 samples and find half of them contain toxins. And there was hepatotoxic, they were more common, but also neurotoxic blooms. But later, in that time when I did my research, the structures of all of the toxins were not known, so we couldn't use chemical methods. That's why we were using bioassays. And later, as you can see, in Germany, for example, huge number of samples were analyzed, but then, in that time, they could use chemical methods to analyze. And it's not only a problem in Europe, this is a worldwide problem. So in the Baltic Sea, there are blooms every summer, and they are toxic every time we have sampled this kind of scums seen here. The major producer of toxins is nodulary. You can see that it contains these special cells which they are using for fixing nitrogen. It's a nitrogen fixing cyanobacterium, and most of the blooms here are containing nodularia. And also afanitsomen, but we haven't found afanitsomen toxic in the Baltic Sea. But we have also studied this Gulf of Finland and found that there are anabena, which nowadays are called Dolichospermum. The names are changing all the time, so it's difficult to keep track about the name changes. I've published plenty of papers in the wrong names over the years. And we found that in these coastal areas we have this microsystem producing Dolichospermum occurring, and also our colleagues in other countries have found the same, especially in Polish coastal areas. So nowadays the major toxin structures are known. There are these hepatotoxins. Microcysteines are very, very common all over the world, and then nodularin occurs in, of course, Borrelia nodularia. You can decide by the name. And then there are three types of neurotoxins: anatoxin A, anatoxin AS, and saxitoxins. And saxitoxins are known to produce by dinoflagellates as well. And anatoxin AS now the people are changing the name also of these toxins, but I don't remember what it will be. And if you want to learn more about cyanobacteria and what kind of toxins they are producing and how to analyze them, there is a very good book. We had this cost action, which was all European countries were participating, and this was one of the products we made. So this is a pretty good source for information. So when we found these toxic cyanobacteria and could isolate the toxic producers and also know how to analyze the toxins, we started to think about how much these environmental factors affect the toxin production. And here you can see one example about anathena, which is Dolichospermum nowadays. And the more phosphorus, the more toxin was produced. So our conclusion was that when they are growing well, they are producing also more toxins. So it was not caused by the stress factors as was suspected originally. And there has been studies also about the competition between toxic and non-toxic strains, and we came to conclusion that these toxic strains may need more nutrients. So we also have found new toxins. We decided to look for benthic cyanobacteria. Here is these kind of biofilms we heard yesterday, and we were scratching those stones and all kind of materials and could isolate plenty of cyanobacteria also growing in the surfaces. And studying them, we found this kind of cyclic peptide with fatty acid, long fatty acid tail. And over the years, several variants were found. And then we did studies seeing if it's excreted or is it inside the cells, and it seems that like microcysteines and nodularin, they are mostly kept inside the healthy cells. And this compound turned out to be, we named it by anabinyl lysines, and it induces cytolytic cell death of nucleated cells, hemolysis of red blood cells, echinocyte transformation, and they are potent detergents of cholesterol containing membranes. So there are still, if you look for new sources of cyanobacteria, you find new toxins. We also do a lot of research. We try to find useful compounds from cyanobacteria, perhaps to be used as a medication. And often it happens that we find new toxins instead. So here, recently when we have studied these cyanobacteria already about 30 years, we start to find compounds which have been found by somebody else before. And here is one example of this scytophysin, which were identified from Scytomena already in 1986. And other researchers found it from nostoc and Sylinospermum, and we identified about 33 structural variants of the scytophysin and found that Anapena, most likely Dolichospermum, now is a new producer. And we also studied the biosynthesis of this compound. So when we studied these blooms, in Finland at least, we often see this kind of picture. Even though it looks uniform here, when you look at it in the microscope, you can see that there is several potential toxin producers present. So the only way to finding out which ones are toxic was to isolate those cultures. And analyze them separately. Very seldom those blooms are formed by one organism, but such occasions sometimes occur. So we also started to isolate cultures from the Baltic Sea and also from our lakes. And by analyzing them, we could see that there are indeed toxic and non-toxic strains of the same species. Toxins are produced by cyanobacteria. We could verify that by producing axenic cultures which doesn't contain anything else than cyanobacterial cells. And since then we have used this culture collection. We have now over 1,200 strains, some isolated, for example, lichen symbiosis and or soil samples as well, but most of them come from the water sources. And we have used those cultures ever since, studying all kind of features of these organisms. So we started to use molecular methods in order to see if we can separate this cyanobacteria. Here you can see there are unicellular filamentous, colonial forming, filamentous again, very nice heterocyte nitrogen-fixing species, all kind of cyanobacteria. Based on the genes we can determine the genetic potential of the organisms. And we can also do phylogenetic comparisons of the organisms, and we can study the genetics and phenotypic diversity. So here you can see example that toxic and non-toxic cyanobacteria cannot be separated from each other. Here are the microcystin, and even though you look at the microscope, you cannot say that this one is toxic and this one is not. So we decided to look for this 16 sRNA genes, which are pretty much used in the taxonomy of microbes. But here you can see, for example, these Planktothrix. There are identical 16 sRNA in non-toxic and toxic strain. So that was not a very good way to separate these toxic and non-toxic strains from each other. So nowadays we know that this cyanobacterial bioactive compounds, toxins and bioactive compounds, they are produced by two ways. There is this non-ribosomal way where enzymes, the genes are coding enzymes which are then putting together this kind of compounds, and this has a fatty acid tail, so we know that it's a hybrid pathway consisting of non-ribosomal peptide synthetase and polyketide synthetase. Then there is this ribosomal pathway where this kind of molecules, they are produced by normal peptide synthesis and then modified later. So here is microcystin synthesis in Anabena dolicospermum. You can see that we need 55 kilobase long gene cluster in order to code for these enzymes. These squares here represent peptide synthesis, these balls here represent this polyketide synthesis. So we know that it's a hybrid biosynthesis system for microcystin. The synthesis starts with this ADA molecule here. And the polyketide synthetases are making this ADA. And ADA is a specific amino acid found only from microcystins and nodularies. Then each amino acid is added, and finally this thioesterase at the end of the cluster makes this cyclic peptide. So we wanted to develop a method to differentiate by molecular tools the toxic cyanobacteria, and we decided to use this microscopic synthetase gene E. The E gene is important because this codes for ACTA and glutamic acid, which are very important for the toxicity of these compounds. And there is not much variation because this part of the molecule is found from all of the toxins identified today. And when we analyzed, sequenced and analyzed the genes from different cyanobacteria, we found that this Anaphena, Microcystis, Plankton, Nodularia, we could separate those. So we could not only say that this is toxic, but we could also identify which one is the toxin-producing cyanobacterium. So here is one example. We analyzed, we used ordinary PCR to identify samples taken from 70 lakes in Finland, and most of the lakes contained potential toxin producers. Even though this was the samples were taken very early. Usually we have these cyanobacterial blooms in August, and with these genus-specific primers, we could see that in that time Microcystis was the major organism, and then Plankton, this was very common. And Anaphena dolichospermum, we don't know that they are usually coming in August, so they were less common. So then we did also analysis. We organized these lakes according to their trophic status. And if you follow this dark color, you can see that it's increasing. And it means that all these Microcystis, Anaphena and Planktotrix, they are occurring in the samples more often when we had eutrophic or hypereutrophic lake. So the eutrophication also seems to affect. So then we also developed this quantitative qPCR assay, and we're using it for Lake Tuusunajärvi. Lake Tuusunajärvi is pretty much studied lake because it's nearby the Helsinki University, so everybody goes there. So we followed 2 years and analyzed the samples, and could show that in year 2001 the blooms were more strong, and we could also separate. There was both Microcystis and Anaphena blooms present, but in the autumn season this Anaphena showed as being more frequent. So when we had this microscopic synthetase genes analyzed, we decided to look for evolution of these microscopic synthetase genes. And here is the result. You can see that we compare it with the 16SR and RBOC1 genes, and you can see that they are congruent, which means that these genes are ancient, and also it shows us that this nodulary synthetase has been recently derived from microscopic synthetase. We know that nodularia contains only 5 amino acids when microcystin contain 7. So there has been a deletion in the gene cluster in order to produce this nodularin. So not by us, but somebody else. I had to evaluate a PhD thesis from Australia, and I noticed that they have found the genes for geosmin and medulla isoborneal. And since I was master degree student, I was studying actinomycetes and this kind of problems, and then I decided that we should also develop methods for detecting this odorous metabolite producing cyanobacteria. And you can see from this, that nostoc seemed to be the one producing mostly this geosmin, and some groups of cyanobacteria also can produce both type of metabolites. But in soil samples, the producers are actinomycetes. Streptomycetes are very well known producers of this kind of metabolites. But cyanobacteria in the water environment are common. So here is the conclusion. We have detection methods for potential toxin producers: microcystin and nodularine, anatoxin A, saxitoxins developed by somebody else than us, cylinospermopsin also produced by other research group, and anatoxin AS, the biosynthesis is not yet known, but it's coming. Their American and Brazilian researchers are studying that biosynthesis, and pretty soon I believe we have detection methods also for anatoxin AS. And then, of course, when we have find new producers, we can develop these new primers. So here are the book of detection methods for and quantitation of toxigenic cyanobacteria. This is also a product of this cost action, as you can see. And this time I also was editor and decided that it's a huge amount of work. I will never ever edit a book in my life. I'm luckily retiring, so I don't need to do that. So conventional PCR is very good when you have a great number of samples to analyze. It's cheap, and that's why very much can be used. And there you can develop a method where we use RNA as well, and then you can see which cyanobacteria are actively producing microcystins. Then quantitative real-time PCR, as you saw from the slides, it identifies the main toxin producers in the lake. Then we developed also chip assay, where we had probes for simultaneous detection of toxin producers based on these microcystin synthetase probes, and then cyanobacteria community, and that probe was based on 16S RNA. And we thought that it will be an excellent monitoring tool, and we patented it and all these things, but the company is using it. And of course it's as good as the probes, so we were hoping to get funding to develop it further, but never did succeed. So then there are these metagenomics nowadays where you can find unknown diversity of this microcystin production, and DGGA was earlier used for the same purpose. So now we are living a genomic era in microbiology, and we also have done research on that line, because we have this axenic cultures of cyanobacteria. But one of my PhD students, Hao, he was the bioinformatician, and he wanted to look this non-ribosomal peptide synthetase and polypeptide synthetase genes in the 3 domains of life. He went through all this in that time published genomes, and he could you can see that bacteria were the most dominant, and archaea and only fungi, only 61 genomes were available in that time. And you can see that in eukarya there is several fungi known to produce important bioactive compounds. For example, those which are used for preventing this kind of when you get a new organ and then your body tries to reject that. There are medication from fungi which you can use. Then surprisingly, we found from methanogenic archaea also this non-ribosomal peptide synthetases, but the others didn't have, and this branch is this eubacteria, not eubacteria, of course, this Euarchaea. And here are the bacteria. Here this kind of gene clusters seem to be very common. Actinobacteria, Streptomycetes are very well known antibiotic producers. Then Firmicutes also Bacillus is producing plenty of different bioactive compounds. Proteobacteria very common, and then cyanobacteria also very common. So these four clusters are the major producers of these bioactive compounds. So there is another study made together with our Parisian colleagues. They have this Pasteur culture collection, and it was decided to sequence more cyanobacteria because you can see this black cluster here. It used to be the one, the cyanobacteria which were in that time in the database, which were sequenced, and they were this very small genome, Synegococcus and Prochlorococcus. They couldn't isolate those cultures, or they couldn't do much about it, and that's why they were using a sequencing in order to characterize those kind of organisms. So from Pasteur culture collection, this red color means that there are new taxons in this phylogenetic tree. And our task was to look at the biosynthetic gene clusters in that. And here are these blue color indicate this polypeptide synthetases, red color hybrid non-ribosomal peptide polypeptide synthetases, green color non-ribosomal peptide synthetases. As you can see, they are very common here, but they are not common among these simple small genome Synegococcus, Prochlorococcus, which are the major organisms, nitrogen-fixing, kind of carbon-fixing organisms in the oceans. They are very ecological, very important. Then here are the other type of so-called ribosomal peptides and also terpenes. These odorous metabolites belong to these terpenes. So as you can see that there is huge potential of cyanobacteria to produce all kind of different compounds. We don't know if all these gene clusters are active, so that's one thing to investigate. And we know only few compounds and these biosynthetic enzymes from these cyanobacteria. So there is yet much work to be done. So here is another example. We many bacteria produce bacteriocins, and we decided to look for our culture collection strains and also these genomes of published cyanobacteria. And you can see that we analyzed these 58 cyanobacteria genomes and found over 100 bacteriocin gene clusters and 209 mutative bacteriocin precursors were identified. And when you look at this phylogenetic tree, all these grey colored cyanobacteria contain these bacteriocins. And the structures of these bacteriocins are mostly unknown. I'm familiar about 2 to 3 papers. The prochlorocin found by van der Donk is one of the best example of the bacteriocins in cyanobacteria. So this was the first genome we made, and we made it, as you can see, Anapena, now it's Dolikoskeum. So, and there was big surprises. Cyanobacterial genome are difficult to close. For example, when people were sequencing microcystin strains, they couldn't close the genome because there is so much repeated elements in the genomes. But we succeeded to use this bioinformatics and how we succeeded to close this genome. And you can see that there's chromosome 1 and chromosome 2. The reason we first thought that this is just simply megaloplasmid, but it contained very important housekeeping genes, and that's why we decided to call it chromosome 2. There are both genes for photosynthesis, for example, in that, and few plasmid. And then we saw that all these microcystins, anapinopeptide, Hassalane gene clusters, they were in this major chromosome. Same was true with these bacteriocin gene clusters and anacyclamide cyanobactine compounds. So cyanobacteria, at least this one, is dedicating lots of its genome information for coding this kind of bioactive compounds. So during that process, we could find this gene cluster which is producing hassaliding. Here you can see the compound structure. It contains also kind of sugar molecules attached. And here is the biosynthesis. It's a peptide synthesis gene cluster. There are 10 kind of tailoring enzymes involved. And when we were then studying our culture collection, we found that they are also hassalinase are produced by Anapena, Cylindrospermopsin, Afamitsumenon, Tolubotrix, and together with our Parisian colleagues, we found also Planktotrix producing these hassalines, and they are antifungal compounds. And we decided to look for the mechanism of action of these hassalines, and then found that this membranes with cholesterol is typical for eukaryotic cells. This hassaline was effectively making holes and lysing those kind of cells. So we also studied these leukemia cells, and it seemed to affect also those cells, human leukemia cells. But then the membranes, when they doesn't contain cholesterols like other prokaryotic organisms like bacteria, there is no effect. So ribosomal peptides, I didn't much mention about them earlier, but we decided to look at these because they seem to contain interesting bioactivities, cytotoxic, multi-drug resistance reversing properties, antimalarial, antimicrobial, antiviral, and allelopathic compounds. And typical for their structures is this oxazolines, zoline and diazolines, then sulfide bridges, and then granular keramyl groups. And the first was isolated from patelamide. It was found out that this Prof didendi was producing this compound. They originally thought that it's produced by this eukaryotic host. Prochloron has not been able to, anybody has not been able to cultivate it, but they made metagenomic analysis and could find the biosynthesis. And since then, several papers came. So our contribution to that study was that we found these anacyclamides, 17 different variants, and they were were just cyclic peptides, no modifications. But then we also find from microscopic strains and blooms these kind of compounds which had these sulfide bridges. And then finally we found this kind of very short linear peptides and could show that they are non-ribosomally produced and they are unprenulated and or methylated. That's why these compounds don't break down easily. So combining genomics and chemistry, you can make some findings. We decided to look for these 4-methylproline containing compounds in cyanobacterial strains. We analyzed several genomes and 30 cyanobacterial genomes contained this kind of 4-methylproline coding gene. Over 100 cyanobacteria strains we screened by PCR and LC-MS analysis and found that most of is a major producer of these kind of new compounds we found. And we could identify these 6 variants of nostovapeptins and nostopeptolide 4 variant new variants. And here you can see the effect these compounds do. Here is microcysteines, and when cells get microcysteine, there are this kind of transporters which brings it inside the cells, and it causes apoptosis. But when we have these nostovapeptins and nostopeptolide compounds present, they block this, and then these cells survive. And we believe that this was the reason when people were doing research using this primary hepatocyte as a studying the toxicity. Maybe this was affecting so that the results were not always clear. So when we have genomes, we can also carry out comparative genomics. Here is one example. We used made a phylogenomic tree using these 31 genes, and we were especially interested about Dolicospermum. The other strain is this Anapena, earlier called Anapena 90, which was isolated from Finnish Lake, but this strain here was isolated from the Baltic Sea. So we wanted to compare them, and it seems that they are pretty much similar in this type of analysis. And the other group of we were interested about was Nodularia spumigena. These 2 strains are coming from Baltic Sea. This is from our culture collection and was sequenced by us. And then this strain came from Brazil. And still all these strains seem to be very, very similar. You can see from here. So by this slide I will summarize 3 papers for you. We also, of course, we have a genome. We can then go for transcriptomic analysis, and we did all kind of physiological experiments, and we were interested about the importance of phosphorus and salinity for this Dolicospermum and also Nodularia, how they affect. And here is first I show you this phosphorus limitation effect to Dolicospermum. Phosphorus is important because this cyanobacteria like Dolicospermum and Nodularia, they fix nitrogen. So that's why phosphorus is the key element. And you can see that when we were analyzing, we noticed that this kind of photosystem 1 and photosystem 2, these genes were repressed during the starvation, but then this BST ABCS gene cluster was induced, and this is the gene cluster which is importing phosphorus inside the cells. And some other things were not affected, like nitrogen fixation, chaperones, and so on. And but there was a lot of remodeling in the genome. And based on this research, we also recommend that if somebody is interested about this phosphorus status of the blooms and cyanobacteria, I think this kind of genes are important to study. Usually only alkaline phosphatase genes are studied in those, but we notice that these genes are very good marker for starvation of phosphorus by cyanobacteria. Then I go for salinity. Since cyanobacteria were occurring in the Baltic Sea, we were very keen on seeing how they stand higher salinities, because in fresh waters there is low salinity. And you can see that there was these lipids and chaperones and transcription were induced in those conditions. There was remodeling of amino acid metabolism and repressed photosystem. Photosynthesis was repressed, and there was no effect to these phosphorus import genes. So it seems that this cyanobacteria Polygospermum face stress. They had to try to protect themselves by perhaps making new lipids for the membranes. So then I go for nodularia spumigena, and in this case we were interested about the effects of low salinity, because this nodularia cyanobacterium is really found mostly in these brackish water environments in the Baltic Sea. And here you can see that what low salinity is causing. We saw that it's really affecting, so that when there is no saline present at all, very huge things happen. This amino acid metabolism, transcriptions, chaperones, translation, all these things were induced. There was no effect on nitrogen fixation, and some of the photosynthetic systems here were replaced. So it seems that nodularin is a real saline environment growing organism. So when we had these genomes from nodularia, we noticed that there are genes, the PHN gene cluster present in this. And we know from the literature that this gene is responsible, gene cluster is responsible for breaking this bond between carbon and phosphorus from this kind of phosphonate compounds. And then organisms can have phosphorus for their growth. In the Baltic Sea, often when you measure phosphorus concentrations, you hardly can detect any phosphorus there. But we thought that perhaps cyanobacteria can use this organophosphate. And when we were looking this literature, we found that many cyanobacterial compounds, many cyanobacteria can have this gene cluster, and maybe they are able to use these organophosphate compounds. Organophosphates are produced by some microbes in the nature, but then there are several artificial man-made organophosphates as well. And we noticed that our cyanobacterium was able not to use only these methylphosphonates, which are natural phosphonates. And here you can see verification from our culture experiments that these genes were present in the culture, and then we went also to sampling in the Baltic Sea and found that they are found from the nature as well. So then we did growth experiments with two strains, nodularis fumigena strains, and we were giving them different type of phosphonates, but methylphosphonates was used by these organisms, and in the same time we could measure methane production. So cyanobacteria are photosynthetic organisms, autotrophic organisms. They don't need carbon. They fix it by photosynthesis. That's why they can kick out this carbon source and keep only the phosphorus for their growth. So people have measured quite elevated methane concentrations in the surface waters of the Baltic Sea, and we were thinking that could it be that these elevated methane concentrations in the Baltic Sea might be due to cyanobacteria which are using these methylphosphonates. So my last story is about cyanobacterial viruses. We succeeded to isolate and characterize this nodularia phage. It's a sieve of weights, and you can see that it contains a very long tail. And we sequenced the whole genome of that. And here you can see Baltic Sea, and we had collected over the years various strains from the Baltic Sea, nodularis fumigena strains, and then we decided to test if our cultures are resistant or if they are if the phages lies them. And you can see that even this area where we were sampling very several days, very close by areas, you can see that sometimes the viruses were effective, but sometimes the same population was resistant strains. And we especially became interested about this phage causing this kind of lysis and liberating nitrogen compounds. So cyanobacteria like nodularia is fixing nitrogen. It's going to these cells, and of course there is happening all the time this that cells lyse naturally because they are old and die. But then these viruses are also one vector to affecting the liberation of these nitrogen compounds. So here you can see, here is with red marked the viruses, and here is nodularia. You can see that we made this kind of 23 or 22 weeks experiment in the chemostat, and you can see that at the beginning those viruses attacked nodularia, and the population was very small. But then this resistance started to show up, and here you can see they were growing very well. And here is this, we were interested about the nitrogen, how much nitrogen there are, and here you can see that this Synechococcus put it to grow in the same bottle, and you can see that at the beginning, because these nodularin cells were lysed, they could grow very well, but when this resistance appeared, the growth was less, but still they got some, because this medium didn't contain any nitrogen source. So here you can see that that when the cells are resistant for phages, there is not much nitrogen in the use of the other organisms we used, for example, chlorella and Synechococcus are the several strains, and Synechococcus and many others. And you can see that they are not growing so well because they didn't have so much nitrogen present. But here, when they realized these cells were realized by phages, the growth was much better. We were using, we were growing nodularia first, and then together with viruses, and then use these filtrates to grow in these organisms. So here is the conclusion: cyanobacteria fix nitrogen. The cells can lyse and liberate nitrogen. Then also phages affect for this liberation. We have this nitrogen pool which can be used by the other organisms like Synechococcus, and also then zooplankton and even fish. So cyanobacteria are important primary producers, especially in ocean. They are responsible for most of this carbon fixation, but also in water environment elsewhere. And as you can see, also nitrogen cycle, when they are fixing nitrogen, is important. And I've shown you that this kind of culture collections are valuable resources. We spend a lot of time isolating those cultures, but now we have later on benefited about it a lot, because we have always cultures we study further. Cyanobacteria produce various toxins and new bioactive compounds. I only show you a very few compounds in this presentation. And we know now that these ribosomal and non-ribosomal biosynthetic pathways are used by cyanobacteria to assemble this kind of peptides. And toxin biosynthesis, when we discovered that we could develop methods to detect these potentially toxic cyanobacteria. And I believe that in future, we combining this genomics and other omics tools, including metagenomic analysis and metatranscriptomic analysis, we can combination with the bioinformatics, physiology and ecology research, we will get still much new information about these wonderful organisms, cyanobacteria. So my name was in the first slide, but here are the persons who have done the work. I have had wonderful PhD students over the years. The dots here means that I couldn't remember all of them while making this. So there has been much more, and there has been key persons. Jouni Jokela, he does this chemical analysis. Matti Wahlsten running this LCM facility, KEYNOTE facility nowadays. Doctors David Fuhr and Leo Rouhiainen, they were focused on this, analyzing this biosynthetic gene clusters. And by the way, Leo Rouhiainen, who is now retired, is my husband. So I was ruling him home and also in work. And I have mentioned only few collaborators here, but over the years we have had wonderful collaborations with people from everywhere. And the most funding source for me has been the Academy Finland. I was 10 years academic professor, and this is the research-based professorship. You could concentrate on research only, and it's much wanted. So I was happy to get it twice. And after 2010, I became an ordinary professor, and then a huge amount of teaching and all other issues. And we had these research centers of excellency paid by Academy of Finland. Now I have funding from this Jane and Aatos Erkko Foundation, and then we have this Nordic research center of excellency called NordAqua. And over the years there has been, I think, 11 or even more EU projects where I have collaborated with many people like Dan also was involved in some of the projects. So thank you very much.
So Kaarina, thank you very much for your presentation and interesting slides. And where I learned is that the cyanobacteria, I always saw some pictures, but it's really a wonderful species, looking very nice on the pictures, very inspiring.
They are beautiful.
Yes,
you look at
really
in the microscope.
And what I also learned that we have the same birthday.
Really?
Yes, it's also the 23rd of August. So it's a very nice coincidence. So are there any questions? Yes, please come.
Thank you very much for a very nice presentation. So do we know actually that what physical or chemical gradients affecting toxicity of the microcystis or production and toxicity, but specifically of the
In our studies, when we were studying these strains in laboratory conditions, and we were making all kind of we investigated light, phosphorus concentration, nitrogen concentration, and all kind of different parameters. We could show that whenever these cyanobacteria grew the best, the more toxins they were producing.
Okay, any other question?
Yeah,
Yes,
I
please.
have two more. I have. So, well, starting from like 2010, 2009 from China, and starting from 2012, starting the nitrogen, also external anthropogenic nitrogen, also starting to be involved somehow in the scientific domain. Like before, it was more like a phosphorus, but nowadays it's also the anthropogenic nitrogen was also started to be monitored, and it's getting important. So, what would you recommend in terms of a management practice for example, lakes or that you are working to monitor or to how to identify? Because what kind of gradients shall we look for? Temperature or the nitrogen, phosphorus, their ratios, or what?
I think we all know that due to irrigation, which is causing this kind of cyanobacterial blooms, there are both nutrients are important for microcystis, argonauts, for example, and plankton as well. They are toxic producers, and since they don't fix nitrogen. But then we have often, usually in autumn season, we see this Avanetsemanum and Anathema dolichospermum appear because the other cyanobacteria have exhausted the phosphorus reserves, and they can then fix this phosphorus and nitrogen reserves, and then they can fix this nitrogen and maybe perhaps using these phosphonates as well to proliferate in the water.
We should always look for the measure or monitor all the time.
Yeah, I think we should try to prevent this nutrient coming, but as I mentioned yesterday, it's very difficult because, for example, we are having huge storms and lots of rainfall, so all soils are washed out to the waters, and that's perhaps the biggest problem now.
My final technical question that I really wonder, because you said that you spend a lot of time for the isolation and for the identifying the species, because so you showed that the amplicon sequencing, and then you showed that actually I think you did a lot of like deep amplicon sequencing because you saw up to the species level that each individual of the name, and which is normally very difficult. There are lots of uncertainties, and you identify 3 or 4 of them sometimes. How could you identify that?
We actually did a lot of taxonomic work. There was one good PhD student, Pirjo Waklin, who was working with Komarek. Komarek is the most well-known cyanobacterial taxonomist, and they clicked together and they did wonderful work together. So, we had cultures, so we could amplify the whole 16 as a region.
So you didn't look at like for the microscope or?
Yes, definitely. With the Komarek you cannot avoid that.
Thank you very much.
Any other questions or comments? Yes, please.
How do you go from screening genomes to a new bioactive molecule to the bioactive function organism or in a membrane, how is that process? Is it very long or is it quite straightforward?
It's very long. I can tell, and you need good collaborators. For the mechanism of action, my PhD student went to a laboratory in Norway. Lars Herfinglad is kind of a person who is studying this amyloid leukemia, and together with him they found out the mechanism of action. And then Jouni Jokela, he when I hired him many years back, he spent 3 years before he learned how to identify this kind of cyclic peptides. Now he is able to understand these NMR results, which I don't understand at all how you can make a compound structure out of this kind of data. But he's very good, and I have been extremely lucky to have very, very good collaborators over the years. Its collaboration is always a good thing.
Any other questions? Yes, please.
I would like to ask one question on the mechanism. So you say there are some important toxic stuff of the toxin, the ADA and the G blue, for example. And I don't know like what is or what are the mechanisms of these toxins that induce the toxicity in the body.
This, I forgot to say that this microcystin and neurolerin, they are protein phosphatase inhibitors, and this ADA molecule, it has been this microcystin has been crystallized together with this protein phosphatase, and they have seen that this ADA goes for this active side of the this enzyme and prevents its functioning.
I would have a question, just adding on that what Karen mentioned. So what I got is that the nutrients is the most important parameter. But is there, and it's very difficult to control them or to get rid of any agricultural influence in all our water systems. But is there any other parameter on which we could handle from the management point as a human influence, like temperature might be a bit difficult, but maybe increase of velocity or changing water levels or amount of water which is exchanged in a reservoir, for example, per time unit? Or is there any other parameter which can be influenced from our side to reduce the cyanobacteria bloom?
I think earlier when they were studying cyanobacteria in the South Africa, they had these dams built for reservoirs, and they were always full of cyanobacteria, those kind of reservoirs. So whenever there is a chance for cyanobacteria to grow, they will. They have studied this Lake Tuusulanjärvi for many years. It has been very productive in the sense that it's blooming every summer, and people have tried all kind of tricks, and sometimes they get something less cyanobacteria blooms, and often it's just cool weather or something like that. So it's very, very difficult.
So if they got less cyanobacteria blooms, it's because of the
weather
specific
conditions.
It's not a specific trick, it's more
they
like
have even taken this smart away from the lake and all kind of things, because there, of course, are these phosphorus coming from the sediment.
Yeah, okay. Any other question or comment? Not at the moment. So then, thank you again.
Thank you.